Good evening, everyone. I hopefully will be informing and entertaining you tonight. And it is a talk that will be based around microbes, many different types of microorganisms, and the things that they can do for us within society. So, I have one of these. Before I start, I better tell you a little bit about myself and the discipline I work in. I'm a biochemical engineer, and what we do generally is we grow cells. We use things that look like this to grow cells. They're called shape flasks. We also can grow them in what we call a fermenter or a bioreactor, and they're just a small stirred tank. The stirred tank can vary from the size of a Coke can up to the size of a very large building. Well, after we've grown the cells, we do some solid liquid separations. That's really just using things like this, which are called centrifuges. They use centrifugal force. They spin things very quickly, and you get a density separation from the liquid and the solid. These are used at a very small scale. At a large scale, a centrifuge looks like that. After that, the next step and the final step of any generic bioprocess is to purify things, and we use chromatography columns, which are just in front of the young lady there. Someone much smarter than me once said that one of the difficult things to do is describe what engineering is. Engineering is a really difficult thing to describe solely because there's so many different branches and fields of engineering. But a way to kind of sum them all up is that engineers make things that make things, and engineers make things better. So as a biochemical engineer, the things we make are actually processes. We're a division of process engineering, so we make processes that will make a product from a biological starting point. The things that we make that make those things could be the equipment within the process, but it also can go down to the individual cell, doing some cell engineering so that you can get a cell that wouldn't normally make something, make the thing you want. And when we say making things better, all we really mean is optimization of the process. So we're looking at either putting less in and getting the same or more out, or putting the same in at the start and getting more out. I specialize in fermentation. There's a reason why I specialize in fermentation, and I think it's the best part of the process. I think it's the best part of the process because without it, you couldn't have the rest of the process, because it's a start point, but also because it's the part in which you need to know the most. As an engineer, I have to know things about the cell. That is the cell itself, so how the cell is made, the structures within the cell, and also how the cell will use things, metabolize things, break things down, make things within itself. But also, as well as knowing things about the cell, I have to know things about how heat is transferred across surfaces, how heat and masses can be mixed and moved. And finally, I have to know a little bit about how fluids move and flow. So within the rest of the process, you might need to know two of them. You rarely need to know all three. So I think the people who work at the fermentation end of the process are the best engineers there are. Fermentation, I think, as I will hopefully go on to explain across the course of the talk, is one of the fundamental parts of society. I think this next slide kind of explains how some people see fermentation and how it really impacts society. The last point being fermentation and civilization are inseparable. This was said by an American poet called John Ciardi, who was a drunk. So that's why he thought it was. But if we go back to the beginning of civilization, and we think about the use of fermentation, there was a bit of a disagreement about whether or not we started to brew beer and wine before we started to make bread. It's actually a little bit before that when the first fermented goods started to appear. Before humans arose, there's the thing called the drunken monkey hypothesis, which says that essentially, some monkeys thought there's some fruit on the ground and it tastes good. It actually, fermenting actually makes things a little bit easier to digest. So what had happened was they developed a competitive advantage by being able to consume the food that had been fermented on the ground, and they got a little bit drunk. Fermentation as a process began, as I think, with most things, in the cradle of civilization in Africa. It's where all good things started. But it really did, so fermentation as a whole, so we're talking here now about mostly brewery and baking, spread across, as civilization spread across the world. A little bit later, fermentation started to be used within the dairy industry for making cheese, and it's a very, very good way of preserving foods. It essentially uses bacteria that are not harmful to get rid of the sugars that could be within the substance that could be used by bacteria that are harmful and could poison you. So it helps you preserve food, and that's why it would have spread and is integral to civilization. So the first fermented goods were found about 9,000 to 10,000 years ago. And not very much happened for many, many thousand years. And that isn't because everyone was drunk. It just did, nothing really happened for a while. Then we had the age, kind of the microscope. So we start at the beginning, around 500 BC, up to the 1500s. The first three people on the list really only had the ideas that there were microorganisms that existed. And then with the discovery, the invention of the microscope, we started to look at smaller and smaller things. So Hooke, with his microflora, Leeuwenhoek, who first saw a bacterial cell, and it's an interesting feat, I think, for civilization to have lasted almost 10,000 years using something without knowing what they were doing. It was very effective in food production, so we're still thinking about brewing and bread. By now, of course, we have got the dairy industry, but we didn't know how it worked. We didn't know why it worked. We just knew it worked. And it worked repeatedly again and again and again. Next on, we're up into the 1700s. So in the 1700s, we have what is termed a mini microbial revolution. You have Edward Jenner with the first discovery, who developed a smallpox vaccine. That in itself, I think, was one of the few things that was a stroke of genius. How he managed to come up with a vaccine with very little information is amazing. I think it probably helped that he didn't initially have to test it on himself, so the risk is mitigated somewhat. But moving on, we have Louis Pasteur with the germ-theory diseases, Joseph Lister, who was the first person to do antiseptic within surgery, John Tyndall, who's some of his work is in front of me today. He was looking more at the ways of removing germs from things. So he came up with the concepts of heat sterilization. 50 years before Fleming found penicillin, he was already trying to work out what it was that was causing clearance zones within agar plates. So he knew that there was something, a substance that could kill bacteria. Then Robert Koch, he was the first person who was growing bacteria within liquid media, which is a massive step change, which is obvious if you're an engineer, a process engineer, but it's much easier to grow a lot of a cell in a liquid than it is on a flat surface. You just have a larger volume and more space to grow things. And moving on all the way through to the late 1920s up to the 1940s, where we have Florey, Chain, and the Fleming kind of combined discovery, where Fleming in the early part, in the 1928s, discovering and working out what penicillin was, moving on to Floyer and Chain, and their effect on society within the early 1940s, with the industrialization of the production of penicillin, the effect it would have had on the war, with the ability to treat injured soldiers who would have normally have lost a limb or could have died from an infection, but were able to be treated by penicillin. And then we think about the fact knowledge is power. As we've already said, there's some wonderful things in front of me. One of the things that would have slowed down progress is an inability to look at things. So with the kind of microscopes that we have in front of us that were created by Faraday, you could see a microbe, but you could only see that the microbe exists. You couldn't see what we could see now, which is the internal workings of a microbe. So we go on to 1953. We have the Nobel Prize for the discovery of DNA, and as is the way now that we know exactly what happened, we can't forget Rosalind Franklin, who was the student who discovered DNA through X-ray crystallography. And this is one of the excerpts from her thesis. But that led to yet another step change in what we knew and what we could do. It's quite a while again, so we have to wait another 20 or so years. Again, this isn't because people were drinking too much. It's more about being able to apply the new knowledge that you've gained. So the next part, I take a little kind of aside and explain something first. So this is kind of a cartoon image of a cell. What it shows is there's two types of DNA within it. So the next step after discovering DNA is working out what we can do with it. What biochemical engineers did was you create recombinant DNA. So what we have on this side is the bacterial DNA, and then over here is the plasmids. The plasmids are what we call recombinant DNA, and recombinant DNA is really just DNA that is taken from somewhere else, formed into a circle, and then stuck into a bacteria. So what it can do, though, is if you pick the right bits of DNA, so for instance, the initial one, which I'm going to come on to next, was for insulin. If you find the genes that produce insulin within the body, you can cut out that genetic information, form it into a circle, stick it into a bacteria. The bacteria will then make your insulin. So the first one, as I've just said, is 1978, a great year, a year of my birth. They discovered how to make recombinant human insulin. It's a massive step forward because before that, for all diabetics, we had to harvest insulin from animals. So we'd harvest them from cattle, pigs, cows, and they were effective, but not. And the insulin from animals is very effective, but it's not as effective as human insulin. So the ability to create human insulin within a bacteria was the first step. It was first made in E. coli. Now other bacteria, yeast can make it. So Pichia pastoris is used to make it now industrially, as is baker's yeast or Saccharomyces cerevisiae. Another example of a recombinant protein is the protein chymosin. That is a protein that's found in hardening cheese, and 90% of all American cheese production is done using this, which has been produced ever since the 90s in E. coli also. So we're getting kind of closer and closer to the present day. What we have to think about is the millennial marvelous microbe. So far, we've been talking a lot about bacteria, fungi, yeast, and molds, because that's what we used up until maybe 30 years ago. From then on, what we've started to do is use different cells, because we have the ability to use different cells. So an algae is a traditional, microalgae is a traditional microbe. What isn't a traditional microbe is a mammalian cell. The mammalian cells usually weren't initially thought to be useful as single-celled organisms, but with this list, what we can see is as you go down the list, more or less, the cells get larger and more complex. With a larger and more complex cell, you can make a larger and more complex product. Sometimes, especially with healthcare, you need a very large and very complex product. So we as a community, decided that it would be useful to be able to use mammalian cells or cells that derive from mammals as the starting block for a biological process. Right, before we get on to kind of the present and future, which is the main bulk of the talk, we need to think about one element, which is a tool which is kind of now ubiquitous within my field of engineering, and that is synthetic biology. I'm going to go through it quickly for two reasons. I don't really understand it, but I do really. But I'm traditionally an engineer, so it took me a little while to get to grips with this. So for a long time, I felt like this, just witchcraft. Wizardry is a little bit of a magic box, and I didn't really know what was going on in it. I also thought to explain it, because to explain it, you actually need to contextualize it. And I thought a good way to explain it would be for me to kind of open the floor to suggestions for things that I could explain how you would make it via synthetic biology. I then quickly realized that would be really, really hard to do. So instead of doing that, I'm going to simplify it. And we're going to go back first to the recombinant DNA, which I've just described, and say, if recombinant DNA is a bit like flat pack furniture, in that you have the bits there, you make the chair, then synthetic biology takes it a step further. What it would do, instead of just having the bits already made so you can make a chair, is it would let you make a chair from a tree. So the tree could be your starting block. The cell would then create the tools to break that tree down and create the pieces that you can put together to make a chair. That is a simple kind of explanation of how the level is increased for our genetic knowledge when you go from recombinant DNA to synthetic biology. But with more knowledge about synthetic biology, so we're talking about some of the stuff that you may have heard of in scientific news, like CRISPR-Cas9, you can go even one step further. So instead of having a tree, which obviously is made of wood, and wouldn't be too hard to make the tools to make the flat pack furniture, you could do this instead. You could use a chair, break it down, make new pieces, make a different chair. You could take a table, break it down, new pieces, make a chair. We're still looking at wood stuff, so it can go even further, hence the picture of the plastic bag. You could literally, as long as it has the right molecules in it and you have the right suite of enzymes, break down anything and make almost anything else. That is the beauty of synthetic biology. So that, I hope, is as clear as mud. But moving on, we now get the joy of looking at kind of the present and the future of each of the things that I started off with at the beginning of the talk. So, the present and the future of fuels. Fuels can be very easily simplified into two different types: gaseous fuels and liquid fuels. So we start with the gaseous fuels first. There are two types of gaseous fuel that people are generally interested in. The first one being methane, the second one being hydrogen. Methane is commonly produced via a process called anaerobic digestion. It's been around for a very, very long time. It's essentially making a process out of what happens within the stomach of a cow or any other ruminant animal. The process itself is quite simply taking some kind of carbohydrate source, using a group of bacteria to break that carbohydrate down into acids, then using a different group of bacteria to turn those acids into a specific acid called acetic acid, which is like vinegar, and then using another group of bacteria to turn that vinegar into methane. It's very simple. It's been done for decades. Hydrogen is also relatively simple. It basically uses any kind of facultative anaerobe, and by that I mean a bacterium that can survive without the presence of oxygen, as well as with the presence of oxygen. And at some point, within its kind of metabolic pathways, it will have hydrogen as an endpoint. Hydrogen production is what I did my PhD on. It's a wonderful process to work on. It's also pretty difficult. It's also very hard to make a lot of hydrogen. But production of hydrogen isn't really the issue with hydrogen usage. The usage really from hydrogen is more about the storage. It's more about societal concerns around the storage and its safety. Hydrogen as a gaseous fuel is way environmentally better than methane because you don't have to burn it. It doesn't produce any carbon, any greenhouse gases. When you use hydrogen to make electricity, you just produce water. So that's the present of gaseous fuels. The future goes back to synthetic biology. As I said, with anaerobic digestion, you have groups of bacteria. The only difficult part of anaerobic digestion is that those groups have to be stabilized. You need to have one group feeding the next group. So you need to be able to feed the initial group at the right speed so that nothing within the mixed microbial flora dies. This could be easily circumvented using synthetic biology. You could put all of the genes required to do every single step of anaerobic digestion within one type of bacteria, and then you wouldn't have the problem of trying to keep a digester stable. With hydrogen, the future, again, would require the use of synthetic biology. What we'd be looking at here is instead of using a potentially useful food source to produce hydrogen, is using synthetic biology to allow the bacterium that makes the hydrogen to use a waste food source to produce hydrogen. Anytime that you can remove a waste from a system, you're always going to be onto a winner. Anytime that you can utilize a waste and create value from that is always going to be positive. So that's the gaseous fuels covered. Next is the liquid fuels. Again, we have two types of liquid fuel. The first one being alcohols. So we talked a lot about brewing, making beer, wine, etc. But you can also use alcohols and burn them as fuels. People don't usually use ethanol, but it is done. But you can use other larger, longer-chained alcohols, and they are effective biofuels. Again, as with the other types of fuel, the step changes here. The future of that field would be to use synthetic biology again to look at what you could use as a food source to feed that process. Another form of liquid fuel is the creation of biodiesel. It's commonly done using algae to produce lipids that can be then transesterified to make biodiesel. That, as a research area, is still alive. The only real issues with using algae as a cell line, no matter what you're using the algae to do, is the amount of algae you can grow. The other problem with liquid fuels, which is why I prefer gaseous fuels in general, is that liquid fuels are by their very nature liquids. We grow cells in water, and water is one of the worst things to have to remove from any system. Water is the best solvent, and therefore, especially when you have an alcohol, which alcohols like water, they're really hard to remove from each other. That separation of water from your liquid can be very difficult, very energy and economically intensive. For that reason, and because lipids, which are produced by algae, are usually non-miscible, so they don't mix with water, it could mean that the future of liquid fuels are more likely to be within the algal and liquid and lipid area. Now, the present and future of pharmaceuticals. This is a great area, and it's the area in which the department in which I work in at UCL does the most research. So the first point being a universal flu vaccine. I guess a lot of people know that the real problem with the current flu vaccine is that we have to work out quite a long time in advance what the likely strains of flu will be. Flu isn't one homogeneous thing. There are many different strains that have slightly different genetic variants. The differences in those genetic variants aren't big enough to mean that you get a different disease, but they are big enough to mean that you need a different vaccine for them. So the universal flu vaccine is really looking forward and thinking to the research area and saying to each other, are there any parts of the flu vaccine that are universal? And if we think about how vaccines work, which I should explain, because it'll make it much easier to go through this bit. Vaccines basically work by your body being presented with something that's foreign and you developing a memory and an immune response to that foreign thing. With a virus like the flu, it will be part of the flu virus, and the virus is like a piece of genetic material that's packaged up by proteins. So all that you do is the proteins that are on the surface of the package are the things that are not natural to your body, so that's the proteins that your body will attack. So if you can find a protein that is consistent across all forms of flu, you can then create a vaccine for that protein and thereby create a vaccine for all types of flu. And the potential future of vaccines for epidemics is, when I first heard of this, a really, really interesting and quite thought-provoking idea. As we've just discussed, even within flu, there are different strains. The strains are created by something that we call antigenic drift. So that's just changes in genetic material. The thought process behind this is that basically, if there is an epidemic whereby people are dying from a viral contamination, there's a very strong chance that before the virus became a killer, there was a version of the virus which is very similar to the current virus that isn't deadly. If you can find the person who got that version of the virus, they will already have an immunity to the current virus. So they'll be immune to the non-deadly and the deadly versions of the virus. So all you need to do then is find out the genes that make the immune response, that create the immune response within that person, take those genes out, stick them into a bacteria or a yeast or a mammalian cell. That cell will then grow the vaccine that creates what you can use then to vaccinate a whole population against an epidemic. The next 3 points, DNA, mRNA, vaccines, stratified medicines, and personalized medicines are really, really interesting. So if we go to stratified medicines and personalized medicines, the difference between the two, and it's really, so the areas are born out of the fact that healthcare is moving forward. We're looking at preventing over curing, but also moving away from the kind of one-size-fits-all model for healthcare. And diseases are obviously very emotive, so if we simplify it a little bit and look at and think about if everyone in this lecture theatre got a headache, we could do one thing, which I could give everybody paracetamol, and the pain might go away. And that's kind of a one-size-fits-all. But with stratified medicines, what we do is we group everyone into certain populations. So we could think to ourselves that if you are under 20 and you have a headache, the most likely cause of your headache could be that you bumped your head. If you're 20 to 40 and you have a headache, the most likely cause of your headache could be stress. And over 40, the most likely cause of your headache could be dehydration, for instance. And then we just look at you, say, you fit into this group, this is how we're going to treat you. And that is the basis of stratified medicines. If you then think even more precise and we look at personalized medicines, we are now going to, instead of saying you are within this group, we're going to say to you, what is wrong? How do you think you got your headache? Have you hit your head? Are you dehydrated? And ask a lot of questions. So we get a more personalized, precise delivery. So now, if I think back 10 years or so, I was a firm believer that personalized medicines would not work. Which is a little bit like being like going back a few more decades and being a computer scientist and saying, yeah, the internet will never take off. It is very clear now to me and to the rest of the field that personalized medicines are going to work. They're very, very useful. And I'm going to give you a few examples of them to show. So the one that is not up behind me, that is often in the news, is CAR T-cell therapy. So T-cells are one of your immune cells. Essentially what you do is take the T-cells from a patient who has cancer. You then grow those T-cells up, so you expand them, so you get more and more T-cells, so you can use those T-cells as a medicine. The CAR part is an acronym. It stands for chimeric antigen receptor. What that does is essentially chemically conjugate something to your T-cell so that your T-cell will recognize a cancer cell. It then becomes very targeted towards your own specific cancer, and it's your body's own cell, so your body will never reject it. And that is a very, very effective cancer treatment. That is what we would call a cell therapy. There are also gene therapies. Some of the examples that are up on the slide behind me are ones that are actually I've just seen over the last week or two in the news or within the scientific news. So up in this corner, we have the pumping heart patch. It hasn't yet been implanted into humans, but essentially what they have done is taken heart cells from a person, grown them up to create a patch so that it can fix any damage in the heart cells of any heart attack patient. Even more amazing, I think, is the use of gene therapy. So this one over here, which is actually a little older than the current state of play, it uses viruses to deliver DNA. Essentially, you grow some cells, you get those cells, you infect those cells with a virus, the virus proliferates within those cells, you take those viruses out. Within that virus, you have packaged some DNA, and the virus is used as a DNA delivery vector or DNA delivery mechanism. The beauty of this is it can be used to solve genetic diseases. So this one is an example whereby it is used for cystic fibrosis. So this is an example where it's used, I think, as an inhaler. So you can inhale a virus. It will then change the cells within your lungs. So it produces a protein, which is the thing that is deficient in people with cystic fibrosis. This can go on even further because it can be used with the right delivery. So this is also linked to DNA and mRNA vaccines. With the right delivery vector or with the right delivery method, you can solve many, many, many different genetic diseases. We're staying away from genome editing as a subject of the talk, mostly because it doesn't involve growing any cells, really, and growing cells is what I do. And before I move on, I should probably also say, if anyone's interested in growing cells, it's a great job to get because cells grow, that's what they do. So I never really have to do anything. So looking forward, again, we're looking at the present and future of food. I think most people will be aware that food has changed over the last 5 to 10 years. We are starting to revitalize old processes. There's lots of fermentation-based processes, so we're looking at people realizing the beauty of fermentation as a preservation method, creating things like kombucha as an everyday beverage, and that kind of revitalization of old processes is one of the things that I really, as a person, enjoy, because I like to see that fermentation. I generally like to see the democratization of knowledge. So I like to see the fact that now we're in a world where anyone can do some form of fermentation, and people do. So it's from kombucha to people making their own yogurts. There's a great and a wonderful growing craft beer industry. Then also baking, the revitalization of sourdough and baking within kind of everyday life. So one of the things that we need to think about in fermentation and the present and future food is basically because we are within, well, for now, the EU, we can't use synthetic biology. There is no law within the EU that allows us to do any kind of genetic modification for our food. The law's different in the States. So some of these examples that we got up here are very US-based, but we can't do here any synthetic biology, which is nice. It makes things a little bit harder, but it gives us a new challenge. One of the biggest things, I think, for the future food will be the creation of new protein sources. And some of these are examples up here, and it's really just like looking at making new types of meat or meat substitutes. So if we start with meat substitutes, there was a company out in California called Impossible Meat. They essentially use a plant-based protein. Doesn't interest me because you don't grow it in the same way that I would grow things. You obviously have to grow the plants. But what they do to make it taste like meat is they grow yeasts that have been genetically modified and get that yeast to produce heme. Heme is one of the things that's within meat that makes meat taste like meat. So you then add that heme into the reconstituted plant protein to make what is apparently a very good beef substitute. I won't know until I go to the States because it's genetically modified, so you can't get it here. Again, moving on, cellular agriculture is a wonderful area of research. It actually goes back to cell and gene therapy, so it's one of those things that I thought would never work. But there is a company that makes it, so I am wrong. Memphis Meats, they make both a beef and a chicken substitute, where the beef and the chicken are both grown from cells of beef and chicken. The reason I thought it wouldn't work is because I was thinking about things like steak. And most of the things that, when people were talking about cellular agriculture, was like lab-grown steak. And I was like, that can never work because you'd never get the structures correct because steak has got many different types of material within it. It's not just one meat. And to try and grow that would be virtually impossible. Obviously, we have massive advances with additive manufacturing, so 3D printing, and with the resolutions that we may be able to reach in the future, we may get to a stage where you'll be able to grow up different types of cell, so your meat cell, your fat cell, some connective tissue, and print any type of steak that you want. I'd prefer to print it cooked already, so to take one step out, but that is kind of the future of where things are going. And then the last point about the present and future food is really increasing the linkage between food and health. So we need to think now not just about the fact that I like to grow bacteria, but also about the fact that we all like to grow bacteria, and bacteria is within us. So I'm talking about your microbiome, thinking about gut health, the link between your food and your gut health, the link between the ability for you to use what people term probiotics, but introducing new bacteria into your gut so you can improve your health. And then also the link between your microbiome or your gut in itself, and things like your mental health and well-being and other forms of health, wellness. So this is why up here we have this here, the psychobiotics, the future of mental health. There's a lot of research that goes into the effect that your microbiome has on your mental health, and greater understanding of that area should mean that we should be able to utilize bacteria even better to not just help with food and physical health, but also mental health. Now, can our marvelous microbes make or do more? So the beauty of them is actually one of the things that's been the biggest negative about microbes to date. The fact that they evolve so quickly is why we have antimicrobial resistance. That is one of the biggest dangers of microorganisms. Their evolution is why they were here so long before us and why they will be here so long after we are. But the fact that they evolve so quickly also means that they have the ability to do things that we wouldn't have thought they would have. That is how we found, only in the last couple of years, strains of microorganisms that can degrade plastic. And it's that ability to generate new genetic material through the fact that they double and their lifespans are so short, they can get through many, many generations really quickly. That is something we will in the future have to use to help us solve society's problems. Thank you.